Awesome. So we are recording. Welcome, everybody. I think this program is mostly just stream keepers. This is our second training session of the year. We had construction site monitoring in February with the help of River Network. And tonight we have finally have the Save Our Streams training tonight with Isaac Walton League. So two of our great partners with the DRWI, we're happy to have them and able to provide the next level of training because of these partners. So thank you guys for joining us. Happy to see you again. I wish it was in person, but it's nice to see some friendly faces again for some Streamkeeper training. Yeah, we already did the recording. We're going to do chat throughout the night. So if you have a question during the program, simply type it in the chat box and we'll get to it either at the end, or if it's pressing, we'll just answer it right then. We have a small crowd tonight, so we should be able to answer all your questions. I think that's all the housekeeping. Try to stay on mute if you can during the program so we can listen to Samantha. And I think that's all I have. I'll keep admitting people during the program, but I'd like to welcome Samantha Briggs from the Isaac Walton League of America, and she is the Clean Water Program Director. She helped us set up this training last year, but it didn't work out because of COVID. So here we are doing a hybrid version of this program. So thank you very much, Samantha. We appreciate your time, effort, and knowledge and passion for all of this. So I think that's all I have. Thank you, Stream Keepers. And if you didn't hear me before, get your names in the chat. And I'd like to introduce Samantha.
Thanks, Kevin. Hi, everyone. I'm Sam Briggs. Like Kevin said, I'm the Clean Water Program Director for the Isaac Walton League. And I was chatting with Kevin earlier. I actually got my career started in the Philly area working at the Schuylkill Center for Environmental Education, and then ended up down here in Maryland as part of the Isaac Walton League, and I've been there basically ever since in various positions. So you're here today to get part one of our Save Our Streams training. Generally what we do is we do this in a single day, and this part would be done in a classroom. But given the current conditions with COVID, we might as well do it online, keep everybody safe, not breathing the same air, and then the outdoor parts will be, distance and learning together out there. So today we're really going to run through the introduction to aquatic macroinvertebrates. I'm going to give you a little bit of background on Save Our Streams and also what to expect moving forward as far as certification and the training that we'll have on April 24th. But today we're mostly talking about the bugs. So it can get a little bit tedious. So, if you need to stand up and do a jumping jack or two during the middle of this, I won't judge you. Grab yourself, a cup of tea or a baked good to keep you awake. But we're going to be talking through the bugs. But first, just a little bit about the Isaac Walton League. We're a nationwide conservation organization, and our mission is to conserve, restore, and promote the sustainable use and enjoyment of our natural resources, including soil, air, woods, waters, and wildlife. So I'm focused on the Clean Water Program, which is really focused on volunteer water quality monitoring, getting folks outside and doing monitoring for themselves to then advocate for better water quality in their communities. And one other thing, folks, I am monitoring the chat as I'm going through this. It's open for me, so please just throw in your questions as you have them. I'd love to answer them. We also have another version of this exact training with the macroinvertebrates recorded. So I will send around the link to that as follow-up tomorrow, including your macroinvertebrate ID test. So don't feel like you have to absorb every single thing that comes out of my mouth tonight. You'll have an opportunity to view it again, and again at your leisure. What else was I going to say about that? Oh, and also feel free to take as many notes as you'd like. Our ID test is open note, as is monitoring. We would never ask you to go out and monitor without your ID tools. Same thing goes for our test. So feel free to scribble away, sit back and relax. Whatever you need to do to learn for yourself is the way I want you to absorb this training. All right, so Save Our Streams is a program of the Isaac Walton League, 50 years old now. I guess I'm like 51 at this point. I really feel like we lost a year in there. But founded in 1969, and the goal was to educate folks about water quality issues across the country. We actually had a staff person who at the way beginning of Save Our Streams, before the Clean Water Act was enacted, would drive around in an RV called the Water Wagon and educate folks about water quality and pull macroinvertebrates out of the stream. So it's been around for 50 years. It started as really an educational tool, and since then it has evolved into so many things. But our main goals are educating the public about water quality issues and also getting that data into local, state, and federal hands so it can be used for restoration purposes. So Save Our Streams is really centered around biological monitoring. So we look for aquatic macroinvertebrates, which are insects and crustaceans. Aquatic meaning they live in water. Macro meaning they're large enough to see with your naked eye, not to be confused with large. A lot of them are very, very small. You can just see that they're there without magnification. And invertebrate meaning they don't have a backbone. Like I said, I got my start at the Schuylkill Center as a preschool teacher, so I personify everything, and I will refer to these colloquially as bugs. For my science sticklers out there, not all of them are bugs, obviously, but that's just what I refer to them as in passing. So when we monitor, we have 3 key steps. We collect the macroinvertebrates, so we can use a kick seine net if it's a rocky stream, or we use a D net, which is that bag net on a pole if it's a muddy stream. We sort the bugs like with like to help us ID them. We identify them using our books and our tools, and then we count the populations of them. And from there, you can get a designation if your stream is excellent, good, fair, or poor. So it's a really great tool for finding out your water quality as a whole. So why macroinvertebrates? The cool thing is their sensitivity to pollution varies. So some of them, in passing, they could get just like a whiff of a pollution event and completely die out. And others are very, very tolerant and will hang out even if there's, raw sewage pumping through the stream. So because their sensitivity to pollution varies, the diversity that you find can tell you about the health of your stream. They live underwater and breathe dissolved oxygen. So they're more affected by those pollution events than, the animals that live on the surface. And it allows us to see how the water quality has been over a longer period of time. If you're doing chemical monitoring, which I will also bring, our test strips into the field to show you how we use those, you're only sampling, the water in your cup at that moment, and it can change in a second. Whereas the macroinvertebrate populations, you're looking at a full season of water quality issues. So while your chemical monitoring could say that your stream is fine, a bad population of bugs can tell you a little bit more about what's been happening in your stream and your watershed. Oops. So we have a new database. It's probably about 3.5 years old now, that we developed with our partners in the Water Data Collaborative called the Clean Water Hub. And this is what we've been using for our volunteers to get their data in one place and really make an impact. So we built this database so that our volunteers had one centralized spot to put in their data. And then we're also working on expanding this database to include other water quality parameters. So you can really kind of one-stop shop for volunteer water quality monitoring data. And so it's free to use. You can make an account. And then from there, you can enter your Save Our Streams biological and chemical data. So here's an example of what our training site would look like near our national headquarters. It shows you a map of where you are. You can upload a picture of your stream, associate it with an organization. We have, I think, like 83 organizations using the hub right now. And then you can also see your water quality parameters over time. And for Save Our Streams, what's really nice is we have all of our parameters graded in excellent, good, fair, or poor. So it's really easy to spread, the links to your sites around, and the general public will still understand that, the poor values are not great water quality and the good ones, are green and excellent moving forward. So it helps really visualize this over time. It's also nice because, since we started Save Our Streams in 1969, a lot of our water quality data is housed in file cabinets, and that's not necessarily the most impactful way to use this. So this is making it readily accessible so that you can pass on your data to local, state, and federal government, but also just your community members and your partners, and share the results that you're finding. So I will also send around a how-to webinar on how to use the hub and how to get that started, and work with Kevin. If you all want to get an organization in the hub, I can set that up as well. All right, so what's in a name? Aquatic benthic macroinvertebrates. So as I mentioned, lives in water. Benthic actually means they're bottom dwelling, so they're living on the substrate, the bottom of the stream, large enough to see with your naked eye. Once again, not to be confused with large, and no backbone. So as we start to get into this, just know that I will bring all of your ID books and pamphlets. I will bring all of the equipment that we need to do the monitoring. We have downloadable dichotomous keys from the website. We have from our website, iwla.org. I'll send around links to those as well, so don't feel like you have to dig for those. And we also have a mobile app that's free called Aquabugs that walks you through identification as well. So it kind of walks you through a dichotomous key that you would be looking at, except it's in an app form. Really useful if you're like me and digging around in streams sometimes without any ID tools, because you stumbled across a stream just out for a hike and you want to see what's there. The majority of the bugs that we're finding are larvae, and we ID down to order level. And we classify them into 3 pollution sensitivities. So they're sensitive, somewhat sensitive, and tolerant. One of the things I also want to mention is I sometimes will refer to the tolerant bugs as bad bugs. They're not bad. You will still find the tolerant bugs in really pristine streams. So what we're looking for is diversity across sensitive, somewhat sensitive, and tolerant. Tolerant doesn't mean bad, and sensitive doesn't necessarily mean good. We just want to see the whole breadth of them there. All right, so a quick refresher on bug anatomy. I could sing a preschool song, head, thorax, abdomen. Maybe we'll do that in the stream if we have a free moment. But the head obviously is at the top of the bug, the head here. Thorax is what would be the middle part. So what we would consider on ourselves as the abdomen, that's the thorax of the bug. And then the abdomen is the bottom part of the bug. So I'm going to be talking about a lot of different features that we're going to see in the bugs here. We're going to have, plates and gills and prolegs and jointed legs and tails. And so I will call out all of those features so that you can help yourself identify between them. Last thing I'll say about the bugs before we get started is that we have an hour and a half and I'm giving you like 80 slides of bug pictures here. So I'm not going to go and give you the background info of every single bug and what it evolved from and what it eats and when it mates and all of the fun facts that you'd like. Mostly we're focusing here on how to ID them and tell them apart from one another. I'll throw a fun fact in every now and again because I have them, but otherwise we're just, we're focusing on the pieces to help you identify them. All right, so sensitive. Sensitive means they are intolerant to pollution. So they require lots of dissolved oxygen and they have specialized habitat or diet needs. These sensitive critters are the ones that tend to fall off when you get into suburbia or urban areas, just because there's a lot of different, types of pollution coming in, the temperature increases, you start to lose that oxygen, and then you'll lose those bugs as well. All right, so we're going to start. This whole presentation goes in order from most sensitive to least sensitive. Okay, so the first bunch of bugs that we're going through are all the sensitive ones, which unfortunately also means they're the ones you're least likely to see in a suburban area. But hopefully we'll see some of them. So this is a stonefly. You can see here that here's the head at the top by the antennae, then the thorax has those plated wing type things, and then the abdomen is at the bottom. So it has a smooth abdomen, so there's no gills on the lower half of the body, and you can see that there. I like to remember stoneflies are smooth like a stone, and that helps me remember for myself that they don't have any gills on their abdomen that you can see. They have 6 legs, so they're an insect. They have tiny hooked claws at the end of each leg. I also like to remember for stoneflies that they have long antennae, which kind of mirror their long tails. It's kind of a mirror image of one another. There's an asterisk there though, because when you're collecting bugs and rubbing rocks to dislodge them from the substrate, the tails and the antennae are probably the things that are going to get lost first. So please don't base all of your ID on just tails or antennae, because if you lose a tail, you could misidentify a bug. Typically, they're about a 1/2 inch to 1.5 inches when you find them, although there are some giant stoneflies that are pretty rare that can get, 2.5 inches long. I've seen those mostly up in like central New York. I haven't really seen them in the mid-Atlantic too much. Last thing, I'll tell you if any of these will really bite you. So you can be aware when we're out in the stream. Most of them won't do anything. So stoneflies won't bite, don't worry. One interesting thing about stoneflies is I joke that they have hairy armpits because their gill tufts, instead of being on their abdomen, are actually underneath their legs. So sometimes if you put them in a low dissolved oxygen environment, like an ice cube tray, if you're sorting them, or a petri dish, they'll start to do push-ups to get more oxygen into their gill tufts under their legs. So it's pretty interesting to see when you're in the field, but that also means that they're really stressed out. So I would just tally them and put them back in the stream if they start doing the push-ups on you. They can live 1 to 2 years in the water as larvae, but only live a few days as an adult when they hatch out and fly away. They're also very solitary, so you won't find hundreds of stoneflies in one spot. They're solitary organisms and very sensitive. All right, moving right along. So I have set the pace for you all. That's about as much time as we'll spend on any single bug moving forward. But I'll remind you again that this is recorded, and it's not even me doing it. So if you want to hear somebody else talk about bugs too, it's one of my other staff people. This is a caddisfly. So also an insect, just like the stonefly, but you can see already that the body has a very different shape. It has a hard plated head, small thorax, and then a long squishy abdomen and 6 legs focused at the top. They can be free living caddisflies, which are the pictures you just saw, or case making caddisflies. And those are exactly what they sound like. They build a case around them. 6 legs total and 2 hooks at the bottom of their abdomen. And they may be in a case that they constructed themselves out of sand, gravel, or parts of leaves or twigs. Can't really see their antennae when we're in the field, and they'll be about 1/2 inch to 1.5 inches long. And here's some examples of case making caddisflies. So you can see the photo on your left that was constructed out of small twigs or sticks that they then stack together, and they use like a thread, like a spider, to then bind those cases together. The one on the top right, that is a micro case maker's caddisfly. So that's a photo that I actually took through a microscope to help you see how small that one actually is. And its case is constructed out of grains of sand. And then the bottom photo is a adult caddisfly. So that's not what we're going to find in the stream, but that's what they hatch out to be. So there's different species of caddisfly that use different material to build their cases. Some have even built their cases out of like green blades of grass, which is really cool to see. They'll do a characteristic caddisfly motion where they're like undulating back and forth to get dissolved oxygen to their gills. And free living caddisflies, they don't build a case, but they do use the silk, just like the case makers, to hold on to rocks or current in the current. They live 1 to 2 years as larvae. And another fun fact is some artists will actually take those case-making caddisflies and put them in a controlled tank environment and only give them access to like precious gemstones and gold and pearls, and then make them build their cases out of those materials. And they're actually really strong. So then they'll pull out the caddisflies, which doesn't hurt them, but it's probably a bummer to spend all that time building a case just to be pulled out of it. And then they'll use those, which are now tubes, as jewelry. It's pretty interesting. All right, moving right along, still in the sensitive category. This is the mayfly. So to me, the mayflies and the stoneflies look very, very similar. They kind of have a similar body structure, similar length of their abdomen. They have tails, they have the 6 legs. But the key difference for the mayfly is that they have gills on their abdomen. So you'll remember that stoneflies are smooth like a stone, whereas mayflies have those tufted gills. And depending on what kind of mayfly it is, those gills can look very different. So they could look like leaves, they could look like toothbrush bristles. There's a lot of different ways that the gills can look. The nice thing though about when we do our monitoring is we're IDing all of these critters live, which means that you can see them moving and you can understand their movements, and also gills are typically fluttering. So you'll be able to pick those out even if it's a really tiny bug. 3 pairs of legs. I always call them the buff bug because they look like they're flexing. They have like wider like leg segments at the top. And they typically have 3 hair-like tails. So here's another reason why we shouldn't ID on tails alone. The first is that they could break off, as I mentioned with the stonefly, but the second is that there's actually also a 2-tailed mayfly. Just to throw, a wrench into our ID practice here. So here's the example of that. We have the multiple types of mayflies. We have the one on the left, which is a small minnow mayfly, and it actually kind of swims like a minnow, which is cool to see in the water. 2-tailed mayfly in the middle. It actually does have 3 tails, so its name is actually a misnomer. You can see a really tiny 3rd tail right in the center of its 2 tails there. And then we have a burrowing mayfly. So you'll notice they look very different, but they all have that similar structure of gills on the abdomen, the tails, and then the 6 legs splayed out. One time I did a training in Palm Springs, California, and I was talking about mayflies, and I was like, but I've never seen a 2-tailed mayfly, so you probably won't either. And so you can guess what's the first macroinvertebrate we pulled out of the stream. The one and only 2-tailed mayfly. So I don't say things like that anymore. They live as larvae for 2 to 3 years, adults only a few days. The larvae hatch into a winged form, not fully adults, and then they have a final hatch. After their final hatch, though, they don't have a mouth or digestive tract. So they all hatch out in one big mayfly hatch, which is where you'll see, sometimes on bodies of water, you'll see massive hatches of these mayflies. They all have to hatch at the same time so they can all strategically mate at the same time because they only live a day or 2. All right, moving right along, still pollution sensitive. This is a water penny. So this is actually the larvae of a type of beetle. So you can kind of see it has a plated body and then its legs are folded underneath. So they are flat and saucer shaped. They have a segmented plate-like body, and they'll actually kind of adhere to a rock like a suction cup almost. So they use their flat saucer shape so the water will flow over them and not pick them up and pull them in the current. 6 total legs underneath and fluffy gills on their underside as well. And they grow up to about a ½ inch. So they're smaller than a penny. So don't let that name mislead you in their size. They're kind of hard to see when they're really, really tiny because they look like little pieces of leaves. So what I'll usually do is I'll take a step back from the net and look, and you'll start to see them moving along the net, and that's how I can pick them out and ID them. All right, so the adults are terrestrial. They live 1 to 2 years as larvae, and the adults only live a few weeks. So they have strong legs to help them cling to rocks in fast flowing water. All right, riffle beetle. So another type of beetle like the water penny. The difference here though is the adults and the larvae both live in the stream at the same time. The adult water pennies will hatch out and fly away. Riffle beetles stay in the stream and live underwater. So it's possible that you'll find larvae and adults at the same time. And generally, if you find one, you will find the other. Oops. Okay, so the larvae looks kind of similar to the caddisfly that we already looked at. So similar body shape, 6 legs concentrated at the top. There are a few key differences here though. The first is that riffle beetle larvae are really, really tiny, much smaller typically than your caddisflies that you'll find. And then they are also very dark, so generally a really dark brown or black, and their whole body is crunchy. So you can see that they have a plated texture. I've joked with monitors and they're like, how do we know it's crunchy? We're not supposed to like put it in our mouth and crunch on it, right? I'm like, no. To me, you can tell they're shiny and kind of waxy, and you can see that plated structure. They look to me like mealworms, if you've ever had a reptile that you fed those to, except much, much smaller. So up to 1/2 inch, really tiny. They also have this gill tuft at their tail, which is pretty interesting to see under a microscope because it'll like undulate in and out, and they'll pull it up and retract it in their body and push it back out when they need more oxygen. So it's kind of like a breathing pattern that they do with their gill tuft at their tail. All right, so the adults, they are small and oblong, really, really tiny beetles. So once again, these guys are 1/16 to 1/8 of an inch as adults. They will look like small rocks or debris, and you kind of have to wait and spray them with water and watch them walk away to be able to pick them out. 6 total legs, again, visible antennae. The key ID for me, because we're looking for benthic macroinvertebrates, right? We're looking for bottom dwelling stuff. Occasionally a surface skimmer beetle or a water strider or something will get stuck in your sample. And we don't count those towards our metric. So the way I can tell if it's a riffle beetle or something else is I put it in the tray of water, and if it sinks to the bottom and then starts walking on the bottom of the water, you know it's a riffle beetle. If it's scooting along on the top and using its paddle-like oars to swim, then it's not a riffle beetle and we're not going to count it. So they're relatively long-lived. They live as larvae for about a year and live as an adult for 5 years, which is why you'll see the larvae and the adults at the same time. They have that like staggered life cycle. They come out to the edge of the stream and pupate and then return to the water. And the adults actually do have functional wings, but rarely use them. They use them once to find that suitable habitat and then live underwater from then on out. All right, so a water snipe fly. These guys are pretty interesting. They have a squishy segmented body, as you can see, and they look like your stereotypical caterpillar. They have like those little suction cup legs that you would see if you were like draw a cartoon of a caterpillar. Still pollution sensitive, soft fleshy body and tapered ends. So this one is facing to the left. So its head is on the left side and its tails are on the right. They are pale to greenish in color, many caterpillar-like legs, and then their tails are the 2 feathery horns at the back end. So whenever anything is described as feathery with a macroinvertebrate, it's because it's gills, and that's what it's using. You can see them here, they're fluffy. It's called a water snipe because at its head, it has 2 tiny little fangs that it'll poke out. These are generally not big enough to actually pinch us though, so you don't have to worry about that. And I have found water snipes in central New York again, but also in very northern Maryland. So it is possible that we'll see them, although they are pretty sensitive and pretty rare. You may never see one, there it is. Each proleg has a crochet, a ring with tiny barbs that helps them grip. So that's how they're walking along with their prolegs. They retract their heads when they're startled, and they are just not well studied. There's not a lot of folks that have done a lot of work on water snipes, which is pretty interesting. Still pollution sensitive, we have a gilled snail. So, it's a really interesting thing about the snails, but if you pick one up off of a rock and turn it over so the opening is facing towards you, like the image you see here, like the drawing, if that opening is facing to the right, it's a gilled snail. And that's our key way that we ID. Their gilled snails will always have a shell that's in this shape. And then they also have an opening for their shell called an operculum. So they can kind of go into their shell and close a door, so to speak. They don't really get much bigger than an inch. And, you know, they eat using a rasping tongue, which is pretty interesting, and they can live for several years. So these guys are sensitive. Oftentimes in urban and suburban areas, when we're finding snails, we're finding a lunged snail, not a gilled snail, but we'll get to those farther down when we're in the tolerant group. All right, moving on. We've made it through 1/3 of our bugs. We are now going into less pollution sensitive. So the less pollution sensitive critters, they can tolerate some pollution like turbid water or lower levels of dissolved oxygen, but they're not going to do well in really polluted water. So first and foremost, dobsonfly or hellgrammite. They're one of the types of megaloptera. So they have two names. The dobsonfly is the adult. So when we find them in the stream like this, it's actually a dobsonfly larva. Hellgrammite is another name for the larvae. So we'll use those kind of interchangeably. Oftentimes folks would find these as bait for fishing, and that's where they got the name hellgrammite in the past. So we'll use both of those. I usually refer to them as the dobsonfly larva. But what you'll see here is they're pretty much like the dinosaurs of the stream. These are the ones, I'll go back to this picture. These are the ones that will take a chunk out of you. And when I say that, I mean, they can get up to like 6 inches long. They look like a scary sci-fi thing, and they have these big pinching mouth parts that can really get you. I've seen kids cry when getting bitten by a dobsonfly. I've not seen them draw blood personally, but they can hurt. You will never see me like free holding a dobsonfly without like forceps or a petri dish or something. 3 pairs of legs looks like a lot more, but if you look closely, the 3 legs are concentrated at the top. So they're segmented, and the rest of these down here, they're not legs, they're just appendages. So they have a short antennae up at the top here, large pinching jaws, so be careful, and then 8 pairs of fleshy appendages at the bottom. And the key thing to note about the appendages is you'll notice the fluffy gill tufts underneath, okay? So once again, fluffy means gills. You can see underneath that they do have those gill tufts that they use to breathe. They are generally really dark colored, really dark brown, or a black almost. And this says 3 1/4 to 4 inches, but I have seen larger hellgrammites than 4 inches, believe it or not. They have long jaws as adult males when they hatch out of the stream. They're not used for feeding or fighting. They're just used for mating rituals, which is, I find that so funny. The larvae live for 2 to 5 years. They spend a long time in the stream getting really, really big. And then the adults only live for 1 to 2 weeks. And the larvae are a favorite bait for bass fishing. All right, the close cousin is the fish fly. So also part of the Megaloptera order, but the fish fly is a little bit different than the dobsonfly larva. So the first thing to note is that they have those same appendages, but they do not have the gill tufts underneath. So there's your first clue that you're looking at a fish fly. It doesn't have any gill tufts. The other thing to note is they tend to be more reddish in color, but I wouldn't rely on colors alone because it can vary. And then the last thing that I take note of is that they have these really goofy tails. They have like 2 long tails and 2 forked tails at the end of their body there. These tend to be a little bit smaller than the dobsonflies, but they can still get pretty large, up to 4 to 5 inches. And they use their 2 tails that they're actually breathing tubes that they can use to breathe air. So that gives you a clue as to why they're in the less sensitive category instead of the sensitive category. They have another means to get their oxygen. 2 to 5 years as larvae, 7 days as adults. And then the adults actually lay eggs on overhanging vegetation like tree branches, and then the hatch larvae fall into the water. So an interesting way to reproduce there. All right, the last one in the Megaloptera order here is the alderfly. And folks generally lump them in as like those are the big three of the Megaloptera, but I find that alderfly look a lot different than the fish fly and the Dobson fly. So some people say they look like a small hellgrammite. They do have the 6 legs again, but one key difference is they have a long, thin branched tail that I think just looks like a feather. They do have 7 appendages again, but they don't have gill tufts, and they are light colored. So the appendages are translucent. So really almost see-through. So oftentimes I need to get them under like a microscope or into the light so I can see those appendages. All right, Becky just asked, how long can a typical larvae live out of water? I'm thinking of drought events. For drought events, they're not going to live longer than a few hours if they're out of the water. So that's why when we ID, we try and get it done in a couple hours, get them in a pan of water, and then put them back in the stream. With drought events where the water level gets really low, if it starts to dry up on the edges, the larvae move with the water generally. If it's something where the stream completely dries up and they don't have anywhere to go, typically they're gonna die off. And that's pretty normal and something that those populations have survived through. The problem that we'll see with drought events is if it's something like with climate change, if it's happening way more consistently, we could see those populations being impacted. So alderflies, like other Megaloptera larvae, can live several years, but as adults, only a few weeks. The adults generally don't eat, so they're there to reproduce and die, which is a common theme amongst our macroinvertebrates. All right, crayfish, crawdads, mugbugs, crawdaddies, so many names for the crayfish. But most folks are pretty familiar with these, either from having eaten them or played with them in streams as kids, use them as bait, what have you. They have 5 pairs of walking legs, including 2 claws. I think the key way to ID these is they really do look like a little lobster. They can be red, orange, brown, or dark colored. They can be a 1/2 inch to 5 inches. Mountain lobsters, yabbies, so many nicknames for the crawdads, and they live 2 to 3 years. One thing I will note for folks is a lot of folks are expecting to pull out crayfish that are, at least 1 inch long, up to a couple inches big. But I have found crayfish that had just hatched really, really tiny, like thought it was a mayfly, got it under a microscope, saw its claws, and realized it was a crayfish. So do keep that in mind. Still in less pollution sensitive, we have the sow bug. So a lot of folks think that these look like roly-polies or pill bugs or potato bugs. Those are the 3 big names that I've gotten. One of the problems I found with doing these trainings across the country is everybody has a different colloquial name for these. Peter had a question, how do you tell native from invasive crayfish? Long story short, Peter, it really depends on your region, what your invasive crayfish will look like. Oftentimes they're really bright in color, and those are the invasive ones. You can look up through your DNR or your county if they have any info on invasive crayfish and what they would expect you to do with them. My advice for folks is just make sure you're not moving anything from stream to stream to keep invasives in check, especially if you don't know if it's invasive or not. But if you do find guidance about specific invasives like the crayfish, they might say to kill this certain type of crayfish, by all means, you can do that. My advice, just don't move it from stream to stream, especially if you're doing an education event or teaching or something and pull them out and put them in a tank. I've seen teachers who've, then gone to their backyard to let them free, but you should go back to the same spot to put them back in the stream. Anyway, sow bugs. So they look like pill bugs or roly-polies or potato bugs, depending on what you call them. They have an oblong, wide body, 2 pairs of antennae. So they have the long antennae and the short antennae. They're dark brown or gray in color, and they have kind of an armor-like plated appearance. And they will crawl slowly underwater like you would see a roly-poly or a pill bug, like on a log or something. Pretty small, no larger than 3/4 of an inch. Generally, they're a 1/2 inch or smaller when we find them. So related to pill bugs or roly-polies, named because females carry the young in a pouch on the body. So it looks like a sow feeding piglets. And the females brood 20 to 250 eggs and then hold on to the hatched young for 20 to 30 days. So that's pretty interesting for a macroinvertebrate. Scuds. So these guys look like teeny tiny little shrimp in our freshwater streams, still less pollution sensitive. They don't have the same super crunchy covering as a shrimp would. They do have a covering, but it's much more flexible. They have 7 pairs of tiny legs, 2 pairs of antennae, and a lot of folks will actually, if they're new to IDing macroinvertebrates, they will confuse these with the sow bugs. And they kind of have a similar body structure, but the key thing to note is that scuds, I call them side swimmers, and that helps you remember how to, tell them apart. So they will scoot kind of, on their side like a C shape and scoot along in the water to then propel themselves forward really quickly, whereas the sow bugs are going to be walking slowly underwater. They can be really abundant in small streams, typically like muddy streams. We've had a group of Virginia monitors who were in a muddy stream that found over 10,000 scuds in just like 1 square meter net. So tons and tons of them. They're not fairy shrimp, so they're a different kind of thing, the scud. And they're generally, if you're finding a stream with tons and tons of scuds, you're not going to have a lot of fish in that stream that are keeping those populations down. All right, so my personal favorite, the damselfly. So they are also less pollution sensitive. And they kind of have similar structures to a mayfly, and I'll show you why from here. They have the 3 pairs of legs, just like the mayfly, and a long abdomen, but you'll notice that they don't have gill tufts on their abdomen. And they also have visible wing pads, which is pretty neat to see in a larvae. So smooth lower body, no gills, narrow body, and then they have 3 tails that are paddle or oar shaped. So you see from above, because they're shaped from the side, it looks like they're not oar shaped, but the side view, you can see that they really do use those tails like an oar to propel themselves forward. It's really neat to see them swim in like a bucket of water or the stream. You'll also notice that when we saw the mayfly, it lays flat against the rocks, whereas damselflies are actually predators. And so they are up on the rock, ready to hunt for their next meal. So they're looking around. They have, the eyes that they can use. They're trying to find their prey and go swim and catch it. They're a voracious predator with an extendable lower lip. So they actually have this weird jaw thing that comes off of their mouth and can like go forward and reach and grab their prey and pull it back into their mouth. They also can rotate their head almost 360 degrees. And they live for 2 months to 3 years as larvae and a few weeks to months as adults. Key thing to note about damselfly adults is when they land, their wings are folded behind their back, like you can see in this picture here. Dragonflies, when they land, their wings are outstretched. So easiest way to tell them apart when you find them as adults. Cranefly, they probably win the award for like the ickiest of the macroinvertebrates that we find because they are so big and squishy looking. And this is a pretty typical size that you would find in a cranefly in a stream. They are plump, caterpillar-like segmented body, 4 finger-like lobes at their back end or their tail. They have a retractable head, so if you poke them and spook them, they'll pull their head right back in. Translucent, white, milky brown, or greenish in color. Kevin says they're the easiest to identify, in my opinion, and I agree. The digestive tract can be seen as it moves. This says they can be up to an inch, but I've seen 1.5 inches or 2 inches. Some mutant crane flies out there. So they're fun to find. The crane fly adults, they look like giant mosquitoes. So a lot of folks will see a crane fly and they'll call it like the female mosquito or mosquito hawks or mosquito eaters. Those are all misconceptions because the adults lack mouth parts. But they live as larvae for up to 5 years and adults only about 2 weeks. Dragonfly, so similar to a damselfly, you can see that they have the large eyes like a damselfly, and they have the legs that they're standing up, ready to go hunt for their prey. 3 pairs of segmented legs on the upper part of the body. Here's your wing pads, just like the damselfly. The key difference between a dragonfly and damselfly, though, is they have an oval or round abdomen. And so this is about as oval as their abdomen will get. Sometimes you'll have a dragonfly that's like shaped like a quarter. That's how round their abdomen is. They have the same scoop-like lower lip as a damselfly. So it shoots out and captures their prey. And if you're ever really bored one day, they have some great YouTube videos of that, of like slow-mo, really great photography, watching those dragonflies use that to pull in their prey. And they can be up to 2.5 inches. Sometimes these guys will take a chunk out of you, but that's way more rare than with like the Dobson flies or fish flies. Arguably, my most fun fact is that dragonflies are actually rectal breathers, which is exactly what it sounds like. So they pull in water through their back end and have the gills on their insides, and then they can shoot them out kind of like a jet ski to propel really fast from place to place. So that's also fun to watch. Live up to 4 years as larvae and up to 6 months as adults. And, you know, some dragonfly species are really, really awesome. So the green darners migrate up to 900 miles, which is pretty neat for a dragonfly. All right, common net spinning caddisfly. So we already talked about our sensitive caddisflies, which is the free living or the case making caddisflies. This is the common net spinning caddisfly, and the name implies that it is the less sensitive version of the two. So these are 2 different caddisflies that you need to identify. Okay, so that'll help you on your test. The key thing to note here is that they have 3 pairs of segmented legs, just like the other caddisflies, but they have 3 dark plates behind their head. So that's a really easy identifying feature. The other thing to note is they have tons of fluffy gill tufts on their underside, unlike the other caddisflies, and 2 protruding hooks with gill tufts on their tail as well. Also, these guys are what they eat. So if they're eating a bunch of green algae, they're going to be bright, bright green in color. And if they're eating a lot of leaf litter, they're going to be really, really dark brown. So don't rely on color alone when you're IDing these guys. They're called common net spinning caddisflies because they make silk nets or shelters that capture detritus for them to eat in the stream and also protects them. Females lay eggs on the stream bottom and passively float to the surface, and larvae can produce noise by rubbing their legs along their head ridges, which is pretty interesting. And here's the adult, and you can see it once again looks like a moth. All right, clams and mussels. So same thing applies for clams and mussels as what applies for crayfish. If you want to look at your local guidance as far as invasive clams and mussels and what to do with them, by all means. For our protocol, we just tell you to put them back in the stream if you don't know if it's invasive or native. And clams and mussels are hard because it can run the gamut of like endangered, threatened, native, all the way up to super invasive. It really just depends on where you are in your region. So they're usually buried in the stream bottom. Their fleshy body can be found between two clamp shells. Most people are familiar with these either from eating them or finding them, out in the stream. One thing I remind folks is if they're doing any sort of monitoring with children, oftentimes they won't know that if you pry them open, they die. So I've seen pure meltdowns from one kid who pried open the clam and all the other kids ganged up on him because he killed the clam, but he didn't know. So just make sure you're letting people know that they can't be pried open while alive. And they can be really, really tiny, like a grain of sand, all the way up to 5 inches. All right. And we are about on time to move on to our last category, which is pollution tolerant. All right. So these are the bugs that can live in fairly polluted water. But most of them do still need some oxygen to survive. So they're not going to be able to live in like a bin of oil. They need, at least some conditions that are survivable for them. Oops. So first, aquatic worm. These are pretty interesting because they have a lot of different distinct versions of the worm. Sometimes they'll just be, look like an earthworm but end up in the stream. Generally, the ones we will find in the stream look just like the picture here. So they'll be really, really long, fleshy body, tend to be kind of pink or brownish in color. And they will like knot up into themselves. Oftentimes they can be frustrating when we're monitoring because they will weave themselves in and out of the net, which can be hard to get them free. And they could even have a small gill tuft on their tail as an aquatic worm. Here is a midgefly larva. These are very, very, very tiny. Oftentimes I will be doing a training and my monitors will be like, we're done. We ID'd all the bugs on there. There's no more left in our net. And then I'll pick up the net and there will be 80 midgeflies underneath their net that just slipped right through. You'll often see them like dancing in a droplet of water on your tablecloth. So they are a uniformly wide worm-like segmented body. They tend to have a dark head, and they have prolegs, one pair at their head and one pair at their tail. So no true legs here, just prolegs. They have one pair of feather tufts sometimes seen on their tail as well, and they can be up to 1/4 of an inch long. So really, really tiny. It's also important to note that there are multiple kinds of midges. They can be brown in color, cream colored, tan, or they can be red. And so those red midges have hemoglobin in their blood and can survive in habitats without dissolved oxygen for periods of time. So if you find one red midge, I would not panic about it. If you're only finding red midges, that might be an indication that you don't have a ton of oxygen. So you're going to be looking for characteristics like the midge dance. So they're going to be dancing in your water, trying to get oxygen to their tail. And they live for about a month. There's a lot of research done on midges and they're pretty interesting, but their total lifespan is about a month. Here is a black fly larva, a little bit similar to a midge, although they have a lot of key distinct differences that'll help you identify them. The first is that they're shaped like a bowling pin. So they have this bulbous posterior that sticks to the rock and then a skinny center, and they get a little bit more bulbous up at the top. They have a distinct head and usually one proleg right underneath their head, which you can see here. Up to 1/4 of an inch long, they can be brown to black in color. And one interesting thing is you'll usually pick up a rock and see like a bunch of black flies sticking to the rock. And then if you knock them off of the rock in the stream, they'll throw out a thread from their tail and then crawl back to the rock on their thread. So they're pretty stubborn. They want to stay in their spot. High numbers indicate organic or nutrient pollution. Because it increases the food source for them. I've seen the biggest black flies I've ever seen in Iowa because they have so much nitrate pollution there. But they can't survive in completely oxygen depleted or polluted waters. Only the female adult black flies bite, fun fact. And they only spend a few weeks as both a larvae and an adult. Leeches, these tend to give people the heebie-jeebies. Which, it's funny. I never had that. My dad and I used to go fishing in Western Illinois when I was growing up. And one time he got leeches as bait and he turned around in the boat and I had a leech on every finger, just like letting them stick to me. So I'm not bothered by leeches. A lot of people are, though. So the first thing to note about leeches is many of the leeches that we find in local streams do not suck your blood at all. They are decomposers and they eat decaying plant matter. So that can help ease your mind a little bit. The second thing about leeches is if you come out of the water and have one stuck to you for some reason, if they're only hanging on by like one point, you can just brush them off. That's just like their suction cup. If they're stuck to you in a U shape, that means they're suction cup to you and they're sucking your blood. So that's when you have to pull them off and maybe sanitize. But they're usually pretty tiny and they usually don't impact us in the stream at all. Oops. They have a worm-like segmented body that is flatter than it is high. They're soft and slimy. Both ends have suckers, which they use to move end to end like inchworms. So that's how you can see them crawling along. And like I said, blood-sucking leeches tend to be uncommon in streams, and they can live up to 10 years. One time I was monitoring with a group of kids in Rock Creek, which is like the famous park that goes through DC. And one kid came up to me and he was like, look what I found. And he had a bowl with water in it. And he had a leech that was so big in his bowl that I thought it was a rock. And I was like, what did you find? I don't see anything that looks like a rock. And he's like, no, look at it. And he had found this leech like on the inside of like a discarded car tire. So it was truly a mutant leech. It was pretty interesting. Flatworms or planaria. A lot of folks will confuse these with leeches, but they have a couple key characteristics here. The first is that they have a soft body. They literally look like a ball of like goop. They're not as, defined as leeches. They have a distinct triangle-shaped head with eye spots, and they will, while the leeches will like inchworm across the substrate, the planaria will just glide along like a slug. The other interesting thing is that if you cut a planaria in half, then you'll just have two planaria that swim away from one another. They fully regenerate if cut in pieces. All right, lunged snails. So we talked about the gilled snails, where the opening opens to the right. Lung snails come in a couple different varieties here. So the first is that there's this orb snail that you see. These will always be lunged snails. You will never find an orb snail that is a gilled snail. They don't have an operculum, so they don't close the front door. They just pull themselves into their shell, and they can be up to 2 inches. Then we have the other type of lunged snail, which is the pond or pouch snail. And you can see it's the same shape as the gill snail, but they don't have the operculum, and the opening opens to the left. So I always remember right for gilled, left for lunged. All right. So you have made it through, so everybody can take a deep breath. And now we're going to run through some mistaken identities. So I have up on the screen a few different macroinvertebrates that people tend to misidentify. So we're going to run through, and I'm just going to have you all type into the chat if you're still with me, which one you think I'm calling out. So we'll start with the top left. What macroinvertebrate do you think this is? Has a smooth abdomen, so no gills. Gill tufts are found where the legs join the abdomen, so it has hairy armpits, long antennae, 2 tails. Peter says stonefly. Does anybody disagree? Smooth like a stonefly. Perfect. Good. That's why I have those things that help me remember. Thank you, Jennifer. Okay, good job. Love it. So that is our stonefly, smooth like a stone. Remember that abdomen is smooth like a stone. And then I'm looking at those 2 tails that mirror their antennae. Next one, we'll look at the top right. So here we have visible gills along the abdomen, often 3 tails. What are we seeing here? Often 3 tails, not always though. All right, so Peter, Kevin, Jennifer, I'll say mayfly. Does anybody disagree? Good. Okay. So this is a mayfly. Great. Generally 3 tails, can have 2, also can have 0 if they all break off. So keep that in mind. And then the key identifier for me are those gills across that abdomen. Last one here, let's look at the bottom. Kevin, the tails don't grow back, but generally they don't need them, or, you know, if it's like all 3 of them break off and they can't get around as easily, then they're just going to be like a solid snack for, you know, a fish or another macroinvertebrate. The bottom one here, so it has the oar-like tails, smooth abdomen, wing pads that I can see, visible eyes. Any ideas? Becky says damselfly. Peter, Jennifer also say damselfly. Great, you're right. So this picture is a little bit misleading because we're looking at the top down, so you can't really see those oar-like tails, but that'll help you identify in the stream is those oar-like tails for sure. All right, so stonefly, mayfly, and damselfly. Great, good job, everyone. Moving on. So these are a little bit more difficult. We have fluffy gills. I'll start with the left. Fluffy gills under each appendage, typically the largest of the Megaloptera, can take a chunk out of you, really dark in color, can get up to 6 inches long. Damselfly, great, good job. The middle one, the close cousin of the damselfly. No gill tufts though. Any ideas? Long, crazy tails that I always like to point out as well. Oh, I'm like misleading everybody. So I read that as Dobsonfly, not damselfly. So I just gave away the first one. So Helgermite or Dobsonfly is the first one. My bad. Second one. Good. All right, I'm seeing fishfly. Ryan says fishfly. Anybody else want to chime in? I'd also like to remind you all that if this mistaken identity thing is starting to make you panic about trying to keep these straight, remember that you can always have your ID tools. All right. So I'm going to send around dichotomous keys. I will bring ID stuff with me in the field. I'll hand out some pamphlets for you all, like our little trifold guides to bring in the field that are waterproof. So you will not need to do this with your own brain unless you want to. All right, good. So Peter, Ryan, everybody says fish fly. You are correct. And now the last one, this is the one that I think looks least like the other Megaloptera. No gill tufts, long fishbone tail, 7 pairs of appendages. The one on the right, good. Jennifer says alderfly. Peter says alderfly. Awesome. Good job. Remember also with the alderflies is they won't get nearly as big as the fish fly or Helgermite. So you're never going to find like a 4-inch alderfly. That also helps me as well. All right, folks, we're in the home stretch here. This one can be the most difficult for folks because they're all pretty similar. So we're going to start with the top left. So we're looking at a bug that has 1 pair of pearl legs below the head and 1 pair at the posterior. So no true legs there. Can be really, really tiny, does a dance. These guys can also be red, so they have hemoglobin in them. All right, Peter says midge. Does anybody agree, disagree? Midge, midge, good. Becky says midge, Jennifer says midge. You are right, so that is the midge fly larva. Let's move down, so we're at the bottom left now. So this one, it looks like it has 6 jointed legs. Its body looks like it's a darker in color, a little gill tuft at the tail, and then it looks like it's crunchy. The whole body is crunchy. Any ideas? This is another one that's teeny tiny. I'll give you a hint. This one also, its adult form lives in the stream as well. All right, Brian says riffle beetle. Peter also says riffle beetle. Good, so this is the riffle beetle. And I always joke that these guys are so tiny that they'll look like little black commas on your net. That's how small they are. All right, so now we're in the top right. We have 3 plates behind the head, brushy gill tufts under its belly, 2 little gill tuft hooks for tails. These guys will spin nets to capture their food. So they're catching detritus as it rolls through. And sometimes if they're eating a lot of algae, they'll be bright green. Sometimes if they're eating a lot of leaf litter, they'll be dark brown, and everything in between. Good. So we're getting, Susan says, common net spinning caddisfly. So does Jennifer. So does Peter. Awesome. So yes, that is our common net spinning caddisfly. So we're likely to see some of those when we monitor in April. And the last one here is arguably the hardest. So 6 legs, long fleshy body. I'm not seeing a ton of noticeable gill tufts, but I'm seeing 2 hooks at the end, a hard head. Any ideas? Peter says water snipe question mark. No, close though. This one's hard right now, I can tell you, because usually when we find these, we find them in their own case. So this isn't a case making caddisfly, but this is a free living caddisfly. So that the way I, ID this one, when it has a case, it's really easy, right? Because it's the only one that actually does build a case for itself. But if I find a free living one and it looks like a caddisfly, so it doesn't have like the caterpillar body like a water snipe, and it's not really squishy like a crane fly, what I'll do is I'll eliminate the other 3. So I'm like, okay, so it's not crunchy, can't be a riffle beetle. It has these legs, so it can't be a midge, but it doesn't have the plates of a common net spinning caddisfly, so it's probably our free living caddisfly. Does that make sense to everyone? And we'll work on that in the field. And once, you get used to where you're monitoring, and like the critters that you'll find in your stream, you'll find some consistent free livers that you'll then be able to recognize really fast. Right, midge, net spinning caddisfly, riffle beetle larva, and the free living caddisfly. Good. All right, opens to the left versus opens to the right. Let's start with the left one. If it opens to the left, it's a what? Lunged left, lung snail, good. So that helps me remember. Lunged snail, great. A lung snail, remember, can also be when it's that circular orb-like snail too. And then if it opens to the right, what's the other kind? Gilled, great, good job. Lunged and gilled. Good work. All right, so here's who I stole all my pictures from for this presentation, so I have to give them credit, of course. If you all are really interested in these and really want to dive in to the nitty gritty of macroinvertebrates, Stroud has a website called macroinvertebrates.org, and it's so cool. They have like this super awesome photographic technology where you can zoom in like as close to gills as you've ever seen in your life and really like dive into the details. So this is my contact info. I'm going to be sending emails to everybody tomorrow with a few things. Okay, so the first will be just next steps, which I'll talk about right now. I will also be sending you all a link to an online macroinvertebrate test. So you would need to pass that in order to become certified. I found, that there are a lot of adults with test anxiety. And so I want to help mitigate some of that from you and not bring you back to high school for a second. It's multiple choice. It's pictures of macroinvertebrates. There's no time limit. And you can use all of the ID help that you need, even to the point of like, you could have this presentation open next to it. So please use any resources that you want to help you. And then it's not a one-shot deal. So if you don't pass the first time, I can walk through it with you and talk you through some of your issues, and then you can take it again. That's part of the reason why we did this session about a month before the actual monitoring. So you do have time to kind of sit with it and ruminate it with it and then pass that test. So tomorrow I'll send you some info on how to do the test, as well as links to all of my resources. And then you'll be able to go ahead and pass that test and then get ready for our in-person training. If you have not registered for one of our field sessions, we have 3 field sessions. Please do so. You can send me an email if you're not on my list to email. And I will help, point you in the direction of that link so you can sign up. We do still have space, so please do. We just want to make sure that we're having folks show up that are registered so we keep our numbers at a good amount so we're not overcrowding. So a couple questions before I keep going. What time of year and what frequency should we search for macros? I try and encourage folks to do it 2 to 4 times a year. Generally fall and spring is the best time. So at least 2 times a year. If you want to monitor more than that, I encourage you to pick up more sites as well. And then the other question is, and before I go to the next question, doing it any more than that isn't necessarily great for the stream, and it doesn't really give you any new information at the particular site. So if you fall in love with macros and want to monitor all the time, just pick a bunch of different sites to get to. The other question, do most of our catch survive after we pull them out of their environment? Yes. So most of them will survive. We put them back in the stream when we're done. A couple things I would want you to be aware of is if it's a particularly hot day, I would try and ID quickly and not have your table in the sun to ensure that they'll survive that, because what's going to kill them is that decrease in oxygen from like the increased temperatures. So make sure that you're getting them back. And if they start to look like they're stressing out, just, tally them up and put them back in the stream. Okay. If anybody has any questions, please fill them in to the chat right now. So just to reiterate, this is, your training part 1. So I'm going to send you a link to your test. I'm going to send you links to ID resources that you can just download from the internet. I'm also going to send you a link to the online trainings that we already have recorded, so you can rewatch this, but with a different person, get a little bit of a different vibe. And YouTube videos that'll help you out, and other resources that have helped me with ID. I'll also point you to our resources for monitors page, which has a bunch of different stuff, including like a guide to clean water advocacy, but also, all of our data sheets and anything that you want to print out. I'll also send you all a link to the Clean Water Hub so that you can start clicking around there and seeing where you'll put your data. Then we will meet for our field session. So just a warning about those. We're taking what would normally be 3.5 hours and cramming it into 2, so that we can get everybody in and out and get the field session that they need to get ready. So if I am coming across as a drill sergeant on April 24th, it's just because I want everybody to get the chance to get in there, monitor, ID some bugs, and learn what they need to learn. And feel free, if you didn't feel like you got all your questions in or wanted to talk to me more about things, you can always shoot me an email and we can set up a time to talk on the phone too. So it's not going to be your one shot to chat with me about this program. So please, for the in-person session in a month, I will send reminders about those too. So don't worry, you're going to hear all of this again, but I'll get it in your head now. I will have all of the gear that we will need. I will have data sheets for everybody to fill out and take home. I will have ID tools. The only thing that you will need to bring is either, a pair of boots or waders or shoes that you can get wet. Those will all work. And whatever you need to be comfortable. So, water, a snack, dress for the weather, as long as it's not like monsooning or, it's another derecho or something, we will be doing this program. Okay. So if we're going to have a little bit of rain, please wear a rain jacket or whatever you need. And then please wear a mask. Because I'm going to keep everybody as spaced out as possible, but there may be an instance where we have to pick up a net together and be within a couple feet of one another. I think that's the gist of it. Please be on time. It will be a quick in and out to get everybody the experience they need. So I wouldn't want you to miss the beginning of it. So to reiterate, sending around the bug test, you have to pass that. Then we'll do our in-person training. After that, you have one more step, which is the protocol test. And let me mitigate anxiety once again for my test anxiety people. The protocol test is just all about filling out the data sheet, and all of the answers to it can be found in either the data sheet or the instructions to fill, doing the monitoring. So you will have everything you need. It's literally reading comprehension. Even, as you're taking the test, if you have a question, reach out to me and I can answer it for you. Because all we want to know with the protocol test is that if you forget how to do the protocol, that you know how to find out how to do it later. Because if you only monitor every 6 months, there's enough space in between. I think that's basically it. If anybody has any questions, my contact is right here. I'm unfortunately on email all the time, so I will get back to you. And I'm looking forward to training in a month. I think it'll be a good time. And stay tuned for an email from me. You don't have a time limit on those tests, so please just do them at your leisure when you're ready for them. And I will nudge you all if we're getting up close to that April date and I'm not seeing tests from all of you. Any last minute questions for me? Feel free to type them in or come off mute if you'd like.
Oh, thank you, Sam. I thought that presentation was great. I've heard macro presentations many times, and you made it very light and entertaining and understandable. I loved how you did that at the end with the comparisons, because that always gets me at the end. And like you said, it's so far and few and in between that you actually get to do this, unless you're Peter or Ryan Newman, who are always flipping over rocks. You don't get to do this that often, so it's easy to forget. So thank you for making an overwhelming topic understandable and enjoyable. That's not easy to do, so thank you.
Yeah, of course.
And for April, I think there was only 1 or 2 people I saw on this list that are not signed up for a time slot in April. So like Sam said, get signed up for that. We will be meeting at the Pennypack Trust parking lot or at the Paper Mill Bridge if you know how to get there and just want to meet us there at your time slot. Otherwise, we'll have a staff member waiting for you at the parking lot to guide you to the sampling site. So that's all the notes I have for April 24th. I think I saw some questions come in here. Let's see. Oh, just one from Jennifer. Kevin, are there sensitive macros in Pennypack? Yes, there are, but not as many as we would like. Occasionally, you'll find a stonefly, very occasionally. We have mayflies, we have caddisflies, we have mud bugs, crayfish. So those are some of the more sensitive ones, I'd say. It's rare. It's kind of rare to find a stonefly. You'll almost never find a helgermite. I've never found one. And we have damselflies. We have dragonflies. So yeah, that I think that's the extent of it. It's rare to find a stonefly, but yeah, we do have some sensitive, and we're seeing more and more as time goes on since the creek's been cleaned up.
That's awesome. Should be.
And we're doing research on that right now. So Maria Paula, she's our research and restoration coordinator, and Peter has also helped out with some research that we're doing. So we're doing active research on this across the board. So hopefully we'll have new and exciting news year after year as we start tracking this. And that's where you guys come in. We're really excited to have some volunteers to help us with this because it's time intensive and it takes a skill to do this. So we're excited to onboard some new stream keepers to help us out with this year. Well, I don't see any more questions, so thank you all for joining us. And like I said, or Sam said, if you have any questions, reach out to Sam, myself, or Ryan Newman, and we'll help you figure out your questions. So thank you, everybody, and we'll see you in April. If you're not signed up, sign up by the end of this week or up until April, really. But it will sell out. So thank you, everybody, and we'll see you soon.
Thanks, everyone.